Great. Samantha. Samantha's a University of Washington. Congratulations, she's graduated, and she worked in the Kalume laboratory. So come on up, Samantha.
So, hi everyone. As was mentioned, my name is Samantha Hanson, and I had the pleasure of working in the Kalume lab this summer at Seattle Children's Research Institute. And this summer, my work focused on the durability of gene replacement therapy efficacy in a mouse model of Dravet syndrome. So first of all, an introduction to Dravet syndrome. It's a severe form of childhood onset epilepsy that occurs in approximately 1 out of every 16,000 births. And it's characterized by treatment-resistant seizures, ataxia, developmental delay, cognitive impairment, and increased risk of premature mortality due to a sudden unexpected death in epilepsy, also known as SUDEP. And Dravet syndrome is predominantly caused by a loss of function mutation in the SCN1A gene, which codes for the pore-forming alpha subunit of the NAV 1.1 voltage-gated sodium channel, as we see here. Here's the alpha subunit. And that's also broken down into the 4 homologous domains that constitute this alpha subunit in the diagram above. So our lab has previously developed an AAV-based gene replacement therapy for Dravet syndrome by precisely targeting the GABAergic interneurons of the forebrain. And they developed this gene therapy after finding that introducing a mutation selectively in this interneuronal population was sufficient to create a Dravet syndrome mouse with the seizure susceptibility and premature mortality phenotypes. This SCN1A transgene was delivered using the AAV vector, specifically AAV2 PHP-EB, and a modified enhancer dubbed DLX 2.0 was used to specifically transduce the intended interneuronal population. And since SCN1A is a fairly large gene and AAV has a limited cargo capacity, a split intine fusion protein system was used to reconstitute the full-length channel protein after expression in the host. So you can see part of the SCN1A gene is incorporated into one vector, and the other section of the full gene is incorporated into a second vector, both of which work together to recombine to form the SCN1A gene in the host. And this approach initially showed protection against thermally induced seizures between P25 and P35, and premature mortality by P70. Here's some other results from the initial findings showing a protection against thermal seizure susceptibility in both myoclonic and generalized tonic-clonic or more severe seizures. And we can see that treated mice were significantly less likely to experience a seizure by 42 degrees Celsius. And on the right, we can also see the EEG data for interictal spikes, MC, and GTC reduction in treated mice. And we notice that the treated mice are less likely to record these epileptic events. So my project had 2 main branches, one of which was the durability of gene therapy efficacy after a year, which depends on the formation and maintenance of the circular episomes, as well as the host immune response. And the other branch of my project was alternative methods of improving the excitability of inhibitory interneurons, but today I'll be focusing mainly on the durability of gene therapy efficacy after 1 year. And I should note that this is especially significant as redosing is currently not an option with the potentially severe and even fatal host immune response following gene therapy treatments. And this work is also important as we consider life phase equivalencies, as a 12-month-old mouse is equivalent to 38 to 47 years in a human lifespan. So it's a potentially long-acting treatment. So my work started when the mice had been injected from P0 to P3 with the SCN1A, and the initial TI testing, or thermally induced seizure testing, occurred P25 to P35. And then I picked up this work from P355 to 375 with the 1-year post-ICV TI testing to analyze these mice after 1 year of the gene therapy administration. And here's what the thermally induced seizure or TI test setup looks like. We have the mouse under the heat lamp, which is connected to the temperature controller, data processor, and laptop, so we can manually increase the temperature. So after giving the mouse a few minutes at a stationary period to acclimate to its new environment, we start raising the temperature by 0.5 degrees Celsius every 2 minutes. And then we start cooling the mouse after it's spent its 2 minutes at 42 degrees Celsius or after it has a seizure. And after testing these 10 treated mice and 3 untreated mice, we found that 100% of the untreated mice experienced an MC compared to only 30% of the treated mice. And we noticed similar patterns with the GTC as the untreated mice experience a GTC 100% of the time, while the treated mice only experience a GTC 20% of the time. And I'll note that this is a severely limited untreated sample size due to the premature mortality phenotype, which I also looked at as I evaluated the TI tested litters, so the TI tested mice and their other litter mates. And I found that there's a statistically significant difference in mortality at the end of 52 weeks, as over 80% of the treated mice are still alive at that time period, compared to only 30% of the untreated mice. And then I also looked at the TI testing in more detail and found that at 42 degrees, the treated mice were significantly less likely to have experienced an MC seizure. And we also note the later or higher temperature onset of MC seizures. And this pattern continues with the GTC data, as the treated mice were significantly less likely to have experienced a GTC at 42 degrees and at 42.5 degrees. And we note the earlier onset, as all of the untreated mice had experienced their GTC by 40.5 degrees Celsius, compared to the treated mice only experiencing a GTC after 42 degrees. And I should note that 42 being a pretty high temperature, we don't actually increase the heat lamp after that, because even wild type mice can start having seizures at high temperatures. So it's significant protection. So conclusions and next steps, our gene therapy approach developed by the Kalume lab in collaboration with the Allen Institute shows remarkable sustained protection against seizure and mortality up to 1 year of age. We do need more samples to evaluate seizure susceptibility and mortality data. And we're also planning to conduct experiments with different controls, such as the vector alone without the SCN1A transgene, or a single vector bringing just a section of that SCN1A transgene to offer better controls for our experiments in the future. Our findings show that this gene therapy approach can serve as a promising future treatment for Dravet syndrome. And further studies will be focusing on the expression of NAV 1.1 in 1-year-old mice, as well as protection against other Dravet syndrome phenotypes with this gene therapy. I'd like to say thank you to everyone who supported me in this program, especially Dr. Ellen Bogan and Dr. McDonald, and thank you to everyone else involved in the program for making this incredible experience possible. And a special thank you to Dr. Kalume for its outstanding mentorship, and everyone else in the Kalume lab for your support through the experience. And thank you again to the donors, to Seattle Children's Research Institute, and to my fellow summer scholars. It's been an incredible experience.